Okay, so today we talk about the hippocampus and sort of how the hippocampus works and what it's connected to, how does it sort of interact with the rest of the brain. And so we'll talk about the basic anatomy of the hippocampus, and then I'll talk about the famous hippocampus patient HM and place cells, and how we can kind of reconcile those two quite different fields. But usually at this point in the year, I have sort of a rant about the limbic system, so the last time we talked about the limbic system, and it's the part of the brain that controls appetite, sexual behavior, drinking, all these kinds of things. And it's really good. It's really good at that. And what's the evidence of that? Well, if you look out in the wild in animals, not animals that have been around people, but if you look at out in the wild in animals, do you ever see like a fat tiger or like a fat deer or fat rat? No, they're all about the right weight. And so how do they do that? They basically have these appetite control systems that regulate their caloric input to within about 1% every day on average. That's about like the accuracy of the system. It's incredibly accurate. In order to sort of not gain weight or not lose weight over time, you really have to nail it right on. If they're a little more active, they'll eat a little more. If they're a little less active, there's a lot of couch potato animals. It's not about exercise. They just exactly regulate their appetite. So what's wrong with humans? Why are humans now 70% of them are obese or overweight? How did that happen? It happened because you're eating weaponized food, and it's designed to basically mess up your limbic system. And how does it mess up your limbic system? It's basically too calorie dense. That's the simple fact of it. It has a combination of salt, a lot of fat in it, and sugar. Those are things that are never combined out in the wild. You have fruit, which is sweet, but it doesn't have a lot of fat in it. And then you've got fatty things like nuts, but they're not sweet and they're not salty. But if you look at the food in a restaurant, or something like, my favorite example is the Dorito. The Dorito is like an engineered, weaponized food, extremely dangerous. You can eat your entire day's calories just in Doritos very easily. And they have salt, oil, and sugar in them. And then they have a bunch of spices, none of which stick out, which have been carefully engineered by taste testers. And what that does is it never satiates you. So if you eat something like ginger, strong flavor, it's attractive, you eat it, and then I'm satiated. If you eat something like a Dorito, it's just got a tiny little bit of ginger, and a little bit of spice, and a little bit of little curry spice in it, and a little bit of this, a little bit of garlic. And you just never get satiated. Plus it's got, the big three, sort of salt, sugar, and oil. And so what happens? Your limbic system still does a pretty good job. It actually still nails it to within a couple percent. So it turns, if you could put on 100 pounds in a decade just by being off by a couple percent per day. That's not very much. That's like, that's like an apple or something, or half an apple. It's not very much. So your system is still really good, but it's just a little bit off, because you're dealing with these weaponized foods. And so what's the answer? The answer is sort of go back to eating real poor people food, which is, who wants to do that? It's kind of insoluble. But what is the answer that's been proposed by sort of large corporations? The idea is sort of biomedical corporations, you'll eat a little chip, and this chip will go in your stomach and transmit out Wi-Fi to the refrigerator. And then it will also monitor your vitals in there, and then it will tell the refrigerator, Dave, you've eaten a few too many bites of ice cream. And so you go to the refrigerator to get the ice cream, and then the refrigerator will say to you, I'm sorry, Dave, I can't give you that ice cream. I've locked the ice cream drawer. So we want to avoid that sort of dystopian plan and go back to eating more poor people food. That would solve the problem, and then everything will work out. So that's end of my rant so, about the limbic system. But the amazing thing is it's still amazingly good, even given this incredible weaponized food that's designed to completely screw up all the appetite and satiety mechanisms. It still does really good. It's only off by a couple percent, but that's enough, to cause a lot of trouble over 10 years. Okay, so end rant. Let's just talk about the hippocampus. So we'll start with the sort of anatomy of the hippocampus. And the hippocampus, when people say hippocampus, as usual, sometimes it refers to just like a piece of the hippocampus. Sometimes it refers to the greater hippocampus. There's about 15 really distinct parts in the hippocampus, and so we'll go over those and see how they're connected to each other. So where is it first? So if we look at a human brain, so here's a human brain. The hippocampus, so the amygdala is kind of sitting right here. And then the hippocampus is sitting here, and it's all sort of crinkled up. That's why it's called the dentate gyrus. And so if you look at it in an MRI scan, you'll see on the medial wall, you'll see this crinkled. That's why it's called the dentate gyrus, because it's like a tooth. So this is the dentate gyrus, just part of the hippocampus. Now, when you look at it, remember evolutionarily, where does it come from? It comes from the dorsomedial pallium. So it originally comes from this. So that's the original neural tube. And where does the hippocampus come from? It comes from about up here. That's where it comes. So it's dorsomedial. So how the heck did it get down there? Well, there's this giant sort of migration and bending of the temporal lobe around. Now, there's a ton of work in the rat hippocampus. And so for comparison, so that's a human. For comparison, here's a rat. I've drawn it a little bigger than it is in real life. So there's the rat cortex, there's the pontine nuclei, and the fourth ventricle, cerebellum over the top, and a big olfactory bulb. So where is the hippocampus in a rat? It's almost vertical. So if you ever look at any papers on the hippocampus in the rat, which is sort of mouse, which is like where most of the work is, it's vertical. So that's, so this is hippocampus there. And there. And so why is it that way? So it's that way in a rat because the rat temporal lobe isn't as developed. So it kind of started off dorsomedial, like up here, and it kind of rotated about 90 degrees, whereas in human it's kind of rotated all the way around 180 degrees. So this is, human and rat. So there's a lot of recording from the upper part of the hippocampus, partly because it's quite easy to get to. So there's human and rat, or mouse. So what we want to do is cut a section through the hippocampus this way. So cut a section through it like that, and sort of perpendicular to it, and look at what it looks like. So it's kind of, it's elongated this way, so we'll cut through the, sort of look at a view of the tube. So if we start, so where's the sulcus outside? So this is the STS. So that's the STS sulcus right there on the outside lateral surface of the brain. And then this is the lateral sulcus up here, where you have so auditory would be right in the lateral sulcus there. So if we look at that from the side, what we get is something like, so we come out. and then go down into the superior temporal sulcus. So here's this is approximately where A1 would be. There's actually a gyrus there, but I'm just sort of drawing it a little flat. So there's A1, primary auditory cortex. And then you get down to the STS. So this is the superior temporal sulcus. And then that's a big one. And then as you go down further, there's some other sulci that's sort of smaller sulci in there. So a lot of times in a human, you have this thing's called the superior temporal gyrus. So put that in there, superior temporal gyrus. So that's this stuff up here. And then usually they divide it into sort of middle temporal gyrus and inferior temporal gyrus, but the big sulcus is the superior temporal sulcus. Rats don't have that at all. It's just completely flat. So as you continue, what do you see? You basically have auditory areas, higher auditory areas extending, all the way down into the STS. When you get down to the base of the STS, you get some multisensory areas in there. So there's multi-sensory. They have like visual and auditory sort of mixed up. And then when you get onto the lower bank, you basically end up in visual areas. And the visual areas continue as you go around. Let me just make this a little bigger. Go around the bottom like this. So the visual areas continue, and so there's a whole bunch of visual areas, and those are called the infratemporal visual areas. So call these, IT or inferotemporal. Those are the areas that are involved in sort of object recognition. And then you eventually get into another little sulcus here. It's not a deep one. It's called the rhinal sulcus. Rhinal sort of means nose, the nosal sulcus. So this guy is the rhinal sulcus. And that's kind of the beginning you can think of that as the beginning of the hippocampus. The cortex sort of changes structure around there. So there's another division as you get close to the hippocampus. You have something called this will be important when we talk about HM, parahippocampal. Hippocampal. And then if you go a little bit forward, you get what's called perirhinal, which is, around the nosal sulcus. Perirhinal. Cortex. And so those are higher order cortex that are right next to the kind of the beginning of the hippocampus. And then once we get past the rhinal sulcus, then we get into what's called entorhinal sulcus, sort of inside the rhinal sulcus. So let's put that in here. So that's entorhinal sulcus, and it sort of comes around like that. And then it sort of comes around this way, and goes back, something like that. Let me make that just slightly bigger so I have more room for labels. Slightly expanded how big it is. Okay, so it's something about like that. Characteristic shape. Okay, and so the part, so the rhinal sulcus is the beginning of entorhinal cortex, and so this guy is entorhinal, the inside rhinal cortex, and that has maybe like 7 separate parts to it. So if you want to read about this, David Amaral. I have some references if you want to look up the subparts of entorhinal cortex. So, and there's some differences between, sort of medial and lateral direction, but there's also a lot of differences as you go sort of anterior and posterior. So this is the rhinal sulcus again. Okay, then as we continue in, we get to what's called the subiculum. So this guy is called the subiculum. Funny name. And it's got 3 parts in it. And those parts are the subiculum proper, the plain subiculum. And then there's the presubiculum. You don't have to remember this. And then the parasubiculum. So there's 3 parts of this. And there's another subiculum called the postsubiculum, which isn't the subiculum, unfortunately. It's something, we'll talk about that when we talk about head direction cells. It's something sort of behind the subiculum. So we got 3 parts in there, little subdivisions in there. And then we get to what are called the CA fields. And the CA fields are, so there's CA1. I'll say what CA is in a little bit. And then there's a little tiny CA2. This is CA2. There's a big CA3. And then there's a CA4, which is sort of sticking right here. And that one is called, I mean, when you think of the hippocampus, you probably read about the hippocampus, you probably didn't realize what a nightmare of little subparts in there, and they're all different, they're differently connected. So this thing here is called, either CA4 or the hilus of the hippocampus. And then finally we get to the dentate gyrus. And so that's the one that's crinkled up, not in this axis. And so this guy is the DG for dentate gyrus. So that's the one that's sort of crinkly. Okay, dentate gyrus there. Okay, so did we get all the parts? So we got if we start, say, at the rhinal sulcus, we have 7 parts here, 7, 8, 9, 10, 11, 12, 13, 14, 15. So there's our 15 parts of the hippocampus, the greater hippocampus. So approximately how are these connected up? And so if you read in a lot of textbooks, you'll have kind of an overall picture of how they're connected up. And we'll start with entorhinal cortex. So here's, it's got 7 parts, but let's just call it 1 for now. Entorhinal cortex. And one of the first projections, odd projections, I mentioned it before, was that the entorhinal cortex goes to the dentate gyrus, DG. But how does it get there? It gets there by this weird pathway. So normally, like if you're a pyramidal cell, and the entorhinal cortex is hooked back up to the cortex. So there's a pyramidal cell. I put it's upside down because the cortex is flipped around here. And if it wants to go to infratemporal, which it does, it sends an axon out and it goes through the white matter, and then it goes and makes a little terminal in infratemporal. But what happens in the hippocampus, and this was seen, by Ramon y Cajal, long time ago. What happens here is the cells, there's the basal dendrite of a pyramidal cell, and then there's the little apical dendrite. What happens is the axon comes out, and it it punches through the peel surface once, and then it punches through the peel surface again and synapses on the dentate gyrus. So, so it's kind of weird. Like, why did it do that? It just kind of decided, I don't want to stay on the white, this is the white matter here, right here. So it's one of the few times when it just decides to go on a crazy path through the peel surface. And so that's this projection. And so this thing is called the perforant pathway because it perforates through perforant pathway. So it perforates through the peel surface, the top surface of the layer 1. And then you have a projection back to CA3, which is the next one in line. And then that projects back to CA1, sort of skip CA2 there for the time being. And then that goes back to the subiculum. It's got 3 parts, but we'll just call it 1. And then that goes back to entorhinal. So that's kind of the classic hippocampal circuit. So you come in, jump all the way to the dentate gyrus, and then sort of go backwards through these structures to get back to entorhinal. Now, it's known in entorhinal that there's some segregation of the input and output pathways. And so if we look at that, so if you look at entorhinal cortex, so here's entorhinal cortex. I'm kind of, it's actually upside, let's draw it upside down. Okay, so it's upside down. So there's entorhinal. And so there's input coming in to entorhinal, and that input pretty much comes in from, so this is, input. It comes in, sorry, I said I would draw it upside down, and then I didn't. So the input, maybe I'll just draw it right side up. It's kind of confusing. So if I draw it right side up, then the input comes in from parahippocampal cortex, and it projects to the upper layers of entorhinal. So this is entorhinal. Then entorhinal cortex punches through to the dentate gyrus, comes back, and then input, then the return pathway comes back to entorhinal cortex, but that goes into the lower layers of entorhinal cortex. So this is like, from subic. And this guy was from neocortex, like higher visual areas. So there seems to be some segregation of the input and output pathway. So you can come into the upper layers of entorhinal, go here, and then come back, go to the lower layers of entorhinal, and from there back out to the cortex. And so we can go back to the cortex through a different segregated pathway. So entorhinal, it's almost like it's got two subparts to it. So, because you might wonder, like, if we just did all this work and we end up back in entorhinal, what is it doing? But what about the input to entorhinal? We didn't mention that. So basically, this gets a whole bunch of input. So it gets, the cortex right next to it, which is, perirhinal and perihippocampal. That's like the next door cortex. So that's a big input to it. But it turns out it gets input from a higher level visual, so we just say higher level. So higher level, visual, like infratemporal, higher level auditory, like some of these auditory areas that are not A1, and somatosensory. So it actually gets quite extensive inputs from all different parts of the cortex going there, higher level cortex. So that's our basic picture. But of course, that's not right, because one of the things that we're going to talk about is place cells, which are found in these. There's a lot of place cells in this right here, in the CA fields. There are in other places too, but we'll just say there's a lot of them here, place cells. And so this was the general story, and then so somebody said, well, what if we just lesion the dentate gyrus? What happens to the place cells? And the answer is nothing. They're still there. So clearly there's got to be some other pathways. And so it turns out that, entorhinal cortex goes here, and entorhinal cortex goes to the place cells, and the subiculum goes to these guys. So there's all different kinds of projections that can help out. And so by no means is it just entorhinal going to dentate gyrus and back through. So there's a lot of back and forth connections. And when we come to HM, it's very important that entorhinal has all these projections back out. That's the ones right here that go back out to perihippocampal and other areas. So this is the PIA. So that's and this is the white matter down here. So we're oriented. Okay, so that's so there's details. Subiculum's got 3 parts. There's a bunch of parts of entorhinal. Different parts of entorhinal get more visual or somatosensory. I've left out some of the details, but this is a pretty good overview. And so what do people call the hippocampus? They usually call this whole thing the hippocampus. So this, something like that, all the way up to entorhinal. So that's, the quote-unquote hippocampus. And when we talk about HM, turns out they took out even more than just that. But when somebody says, the hippocampus, they often refer to all those things. Okay, so that's our connections. And so what we want to talk about first, which order should we do this in? Should we talk about Olds or physiology first? Let's we'll talk about the hippocampal physiology first. Okay, so what are place cells? Basically, there's cells that fire when the animal is in a certain part of the environment. And I like to, tell the story of James Olds, because James Olds was a guy who basically discovered place cells, but because he didn't have the right theory, he kind of ignored the data in a way. And so this is an example of, if you take a philosophy of science course, people will talk about the idea of theory-driven observation. And so what theory you have influences what you actually observe. And so so who is James Olds? Well, he was the guy that sort of put the electrode in the medial forebrain bundle that showed that if you stimulate the medial forebrain bundle, the animal will just immediately press a button to get more stimulation, because it generates this incredible urge to sort of do whatever that, whatever it was that caused that stimulation. And so he set up just a simple T maze. So he just set up a simple T maze like this. And he was thinking of the hippocampus like the way that I was talking about in the previous lecture, that it somehow mediates between the cortex, which maybe knows where stuff is, or the motor system, which knows how to get someplace, and then these things in the hypothalamus that say, you're hungry. It's a sort of a general monitoring of internal state that says you're hungry, and somehow that information has to sort of percolate back out into the cortex, out to structures that actually know how to find something that will sate your appetite. So he had a rat. So here's the rat tail, whiskers, sort of sitting there. And they might say put the reward in this side of the, let's just do a bunch of trials where the reward is in the left side. And so what he was expecting was that when you get around here, when the rat gets around here, what ought to happen is the hippocampus should sort of leap into action and sort of, signal there's the rat. When he gets there, he has to make the decision point and say he knows that the reward's always in that side. And so somehow that's going to then leak back into the motor system or the striatum or upper level visual areas and tell them, we got to figure out where the food is. It's on the left. So maybe they remembered something about left, but they can't sort of like directly monitor how hungry the rat is. And he found some cells like that actually responded there, but then he found some cells that responded when the rat was just sitting here, the hippocampal cells. And he found some cells that responded whenever the rat was in the right part of the T maze, even though the reward was always in the left part of it. And he found some that were sort of responded when the animal was in the reward place. Then he found some that responded like when he was halfway down the T maze. So he basically discovered place cells, but because he was not thinking of the whole system as some kind of internal inertial guidance system that tells you where you are relative to the environment, as opposed to something that mediates between the hypothalamus and the parts of the brain that sort of know where to get food, he didn't sort of pick up on it. And the people who picked up on it instead were John O'Keefe and Lynn Nadell. And so they eventually got the Nobel Prize for it because they had a different theory. Their theory was that this hippocampus was some kind of cognitive mapping system. And so when they did exactly the same experiments, they basically were able to see. So what was happening here is the brain was trying to tell James Olds, this is the way I work. James Olds could not see it. So that's kind of the importance of, it's not just a hypothesis. When the experiment doesn't work, it's really important to say, maybe the world is trying to tell me something and I just haven't asked the right question. And so I'm going to sort of make a new hypothesis. That's actually, how you actually find out new things. So it's interesting when you look at the history of this. So I learned neuroanatomy in 1980, a long time ago, and I learned about this stuff. It was already known in 1980. And when the place cells first came out, I said, I don't believe it. I don't believe it. The brain doesn't work that way. That doesn't make sense. So I went to the neuroscience meeting. I remember I went to the neuroscience meeting in 1980, and I saw this video. The guy, it was back in the day when nobody had laptops or anything, so it was like a videotape player, giant thing sitting next to the poster, and I watched this video. And what the video was, it was this experiment where they had this arena. So they made this big arena, and so there's the arena, and then there's a rat in the arena. So it's a big arena, so here's the rat like that, not too big, whiskers, tail. And he's running around in the arena, and there's the experimenter, is off to the side, and he's tossing little pieces of rat chow in there. So so it's a hungry rat, and he's tossing little pieces of rat chow into different parts of the arena. And then there's a cue card to sort of indicate to the rat, where he is in this arena, because you would take him out like, so this guy's a cue card. And so that, so the rat sort of knows where it is, because every day you would take it out, put it in there again, and then the rat would sort of be able to orient itself, because otherwise it was just sort of a faceless circular environment. And so what does the rat do? Well, it's a rat, and so there's rat chow, and so it just goes all over the place, just running all over the place. Get the rat chow, run all over here, like that, for like 20 minutes. And so the rat it's an upgraded rat. It's got a little headlight on it. There's a little headlight there, and then there's a camera over the whole thing that's just recording. So there's a camera, just recording where the rat is using the headlight. And amazingly, what you find is the cell only fires when the rat is in a particular place in the environment. So maybe just this place. It only fires when the rat's running through that place. And so this is, the place field. And again, it's hard to believe that there could actually be something like this. But when you actually watch the video, I just watched the video and I just said, okay, changed my mind. I didn't believe it. Now I totally believe it. Just that one video immediately convinced me, because what happens is, I've got place cells operating in my hippocampus right now. And undoubtedly there's some that are right here. And the way they work, it's, if I'm just walking around, talking, doing other stuff, the place cell would just go, and then it'll stop firing. And so, it just likes this place. Doesn't matter if I'm turned this way, it'll work that way too. So no matter what direction your head is turned. And so you just record 20 minutes of data, record the position of the animal, and you get this unbelievably high signal to noise thing. The cell just fires when the animal's in that part of the environment. So this experiment has been done a zillion times, and next time I'll tell you a whole bunch of cool, variations on this experiment when we talk about place cells and head direction cells. But they really are true. That's actually the way the CA fields work. 70% of the cells are like that. Yeah.
Okay, I'm confused. So, how exactly does it work?
Ah.
How does it just how does your know just oh, this place, yeah, I'm gonna fire, and then you walk away. I'm kind of confused.
Yeah. No, it's a very good question. That was my initial feeling. The brain doesn't work this way. So how could this possibly work? Well, it's complicated. So it has to know, there's the cue card, but then it has to take into consideration, I know the cue card is over there even though my head's pointing in this direction, so it can't be just a straight visual signal. But it has to be a visual signal that's been processed so that it knows, actually, I'm not seeing the cue card, but I know there's a cue card here, and so that means I'm pointing this direction, and that means I'm probably about over here. But that's not enough. It has to know, how many steps did you take? So the motor system has to provide input, say, the basal ganglia, which is another input. So this guy has to go in there and tell it, how many steps did you take? I turned around, I saw the cue card, and then I backed up 3 steps, and so I know that I'm further back. It has to integrate information about what direction your head is in, so there's got to be some vestibular stuff coming in there, which there is. Vestibular stuff coming in there. So, yeah, so that's the initial sort of amazement. It's how could this possibly work? But it definitely works. It even works, you can pick up the rat and just go, you can actually sort of map the rat doesn't even have to do the motor stuff. It can actually do it without it. And we'll talk about various theories of how this could possibly work next time. But for now, let's just say it actually, 70% of the cells do this, and they actually respond just in a particular environment, in a particular place, in a particular environment. Yeah.
So for the previous one with the teammates,
Yeah.
so why are there multiple places where the cell fires?
Oh, I see. Yeah, no, this was just one cell. Yeah, this is just one cell. One cell, and this is another cell, not the same cell.
So there are cells responding to different places?
Yes, yeah, the cells tile the environment. If you record enough of them, you can predict where the rat is really accurately. And when they're sleeping, sometimes they play through the place cells. You can see they're imagining going through the maze. You see the place cells being activated in order. So, yeah, so it's a real thing. Like I said, I was just convinced in 5 minutes looking at the video that's actually true. Now, let's go to HM. So HM was the famous, hippocampus patient. So let's jam HM into this little space here. So what does HM look like? So HM doesn't look like this at all. So who was HM? So HM was this guy where they took out the hippocampus on both sides. Henry M. He died now, and he donated his brain, and so they were actually able to section it and look at what the lesion was. And so what did they take out? Well, it turns out they took out, and so why did they do this? Why did they do this? So bilateral hippocampal removal. Hippocampal lesion. So the reason they did this is that the hippocampus, especially the dentate gyrus, is a very active place. And it's this whole circuitry down here is quite susceptible to damage. The cells fire very rapidly, and they're especially sensitive to excitotoxin damage. So if any kind of toxin that causes the cells to fire a little bit more, like sort of a stimulant, can be damaging to these guys. Because they're already so close to the edge, firing so high. And so it's not uncommon for epileptic seizure activity to be emitted from the temporal lobe from these structures here and spread across the cortex. And if we look at the here's the septum, top view. If we look at this, there's the connection to the two septums. If we look at the two fornices, the two fornixes that go to the hippocampus here, like that. So this is the top view. There's a whole bunch of callosal fibers. It's actually it's separate from the corpus callosum. It's called the hippocampal commissure. So hippocampal commissure. So the left hippocampus can get to the right hippocampus, and sometimes epilepsy spreads. If it starts in the right temporal lobe, it can spread to the other temporal lobe, and so that's the one and only time that they ever did this, because once they realized what they did, they said, we're not going to do that again. So HM had this severe memory deficit. It's a complex, severe memory deficit. And what did it look like? I don't have quite enough basal gang. Make that a little shorter there. So it's a complex thing. It took a couple years to figure this out. So the first thing was obvious is that he had some kind of short-term memory deficit. So basically they did the surgery in 1952. So here's the surgery where they removed both hippocampi. And then what they found was if you come into the room and say, hi, my name is Marty. So this is, some event. Let me just put that off to the side. So some event happens, like, introduce yourself. So HM will say, oh, hi, Marty. He'll be able to remember my name. And then if there's some event boundary, so event boundary, like, for instance, you go out of the room and then come back in, he's completely lost it, gone. So he can actually keep in working memory, my name for, a couple of minutes if you don't change, if you just sort of keep asking him and don't sort of like cause any sort of boundary to occur. But if you, as soon as you leave the room, it's just like poof, out of mind, and then it's just gone. And this, you see this happen in old people that start to get hippocampal damage. You'll tell them something, and then 5 minutes later they've forgotten that you told it to them as the system starts to break down. So that was called an anterograde sort of memory problem because, an event happens and then you somehow lose it when you go into the future. But he had something else, which was if you go about 2 years before, so 2 years before, so this is like say 1.5. like 1.5 years before. So before the surgery, he's got retrograde memory loss. So in this region, he has retrograde. So he can't remember stuff that happened in the year and a half before the surgery. And the way they tested this with like TV shows, because TV was by this time, TV was just around. So you essentially did a tracer with the TV program to the entire population, so you can grab a bunch of other old people and test them and see whether they have memory of that. And so he's got retrograde memory loss, not just TV, you can also use other events that happen, historical events. And then memory's okay this way. So over here, long-term memory okay. So he can remember stuff that happened like a year and a half before the surgery, but he can't remember something that just happened like 5 minutes ago if you've got an event boundary. And then the third, so there's 3 parts of it. So he can remember all this old stuff. He's got this gap of about a year in old memories that happened before the surgery, and then he can't form new ones. So what does this look like? So if you look at a standard 1960s cognitive psychology model, what does it look like? You got some input, it comes into iconic memory. Maybe you make like an image, like a simple sort of box diagram. And then you've got short-term memory. That's like if you're trying to remember, my name when I first introduced, or a telephone number or some set of numbers. And sometimes people have suggested, they called that working memory. So, some kind of memory that's sort of active working memory, short-term memory / working memory. And then eventually what happens is you might be able to remember a number, like your own telephone number. Like people don't remember the telephone numbers as much anymore, but in the old days they used to. And so eventually somehow we get to long-term memory. It could be one of these things that used to be in short-term memory, and then we store it and we can get it back. We can sort of bring it back out of long-term memory into working memory and do something with it. So that's kind of like the sort of standard cognitive science model. Now, what does HM look like? It doesn't look like that at all. Not even vaguely. So what does it look like? Well, you've got some input coming in, and it goes to some kind of short-term memory, working memory, which will keep it live as long as you don't have an event boundary. So it goes in there. And then what happens is an event boundary occurs. And what you essentially do, what HM does is he dumps the short-term memory, working memory into the hippocampus. It's kind of an intermediate term memory. Nobody calls it this, but I do. So intermediate term memory. And then what happens, then you're okay. You can recall it if you need to. But then eventually it gets percolated back into long-term memory. But this takes like 1 year. So somehow, because HM has normal memory for stuff that's older than about a year and a half before his surgery, somehow the information that got dumped into his hippocampus before it was taken out, somehow over like a whole year goes back and sticks the memory back into the working memory part of the brain, because you can take out the hippocampus. You can take out the hippocampus and you don't damage it. He still has access to those old memories. So when you phrase it this way, some people say like that doesn't make any sense. How could we store a whole year of memory in the hippocampus? And the answer to that is, well, you're storing the whole rest of your life in the cortex. It's only a year. It's not very long. So that's the way it looks like this circuit works. We haven't said exactly what part of the circuit is doing it, but you've got some kind of working memory sort of assemble what's going on in the current situation, and then somebody changes the context, or you walk outside, or you come in and somebody else joins the crowd. Some kind of a boundary occurs, and then at that point you just kind of dump out the contents of working memory into some kind of temporary storage, and then load up contents of working memory with whatever the new situation is. And then over a very long time, you slowly percolate some portion of those events that you dumped into the intermediate term memory back into long-term memory, so that they're now permanently stored, and you can take this out and it won't affect it. Now, when you look at this, it doesn't even look vaguely like a place cell. So this is rat chow here. Don't forget the rat chow. So what's going on here? So part of the problem is that what they did to HM is they not only took out, first of all, they didn't take out the whole hippocampus. So they actually only took out his amygdala and the front part of the hippocampus right here. So this was HM. So they didn't take the whole hippocampus out, and that was confirmed when they scanned him and then actually sectioned his brain. They could see that that's the part they took out. So they didn't even take out the part of the hippocampus that all the rat people record from. There's a lot of place cells, up in the top part of the hippocampus. Here, that's equivalent to the back of the hippocampus. And so if you look at HM, he wasn't even that bad at sort of finding his way around. So they didn't take out the place where a lot of the place cells are. They took out the front half and the amygdala, but then they also took out part of perirhinal cortex and perihippocampal cortex. So this was HM. And so it turns out the reason that the hippocampal place cells don't look like HM is they're referring to different areas. And it took a number of years to work this out, but what they eventually did was they showed that if you damage the hippocampus, you do not produce just the hippocampus, like the dentate gyrus and the CA fields where the place cells are. You don't produce this problem of retrograde memory loss. Now, demonstrating retrograde memory loss is a relatively complicated experiment, because so imagine how you would do this with a monkey or a rat. You'd have to sort of have some events occurring that are memorable, and then some more events recurring that are memorable, and then do a lesion and try to show that you've damaged the events that are closer to the lesion than the ones that were before. So this is a very long-winded experiment. It takes like about 6 months to do something like this to actually pick up the signal. But what they did was they were able to pick up a retrograde memory loss signal. But it turns out it wasn't the CA fields. So if you want to look like Stuart Zola Morgan, and put Zola Morgan up there, and Larry Squire did these experiments. But basically what they did was they showed, retrograde memory loss. But it turns out that the CA fields and the dentate gyrus, if you lesion those, no. And what caused the retrograde memory loss was, perirhinal, perihippocampal. Perihip. So that's actually where this intermediate term memory is. So yes. And there was a well-known patient. This no, where did this no come from? They had a patient who had a temporary heart stoppage. So it turns out, you can hold your breath for a minute, no problem, your brain won't blow up. But if your heart stops for a minute, completely different, because now blood is not being pumped through the the brain, and so even if you're holding your breath, there's still oxygen coming into the brain from your blood, because your blood can hold a lot of oxygen in it. So if your heart stops for a minute, that's a serious business for the brain, and it will kill a lot of the cells that are these ones that are very sensitive to overstimulation, because they're living so close to the edge metabolically that they can be killed relatively easily. And so there was a patient that had this one, his heart stopped for a minute, he was revived, and he had some sort of memory problems, not serious memory problems. They got his brain, he agreed to donate his brain, and they discovered he completely blown out his CA fields and dentate gyrus. There was no cells there, but most of the rest of the cortex was okay. He didn't have any retrograde memory loss. And then with the animal experiments, they basically were able to show that, you get the same thing. If you lesion the dentate gyrus and the CA fields, you don't get any retrograde memory loss. And the retrograde memory loss comes instead from lesioning the perirhinal and perihippocampal. So here's, 1, 2. So that's pretty much how these two pictures have been put together. They're just actually different brain structures. So what's causing these? So this intermediate term memory thing must be somewhere in perihippocampal perirhinal cortex. And so these perirhinal cortex, of course, receives projections from visual cortex and, the auditory cortex and somatosensory cortex. So there's plenty of ways for the information to eventually get back out into other long-term memory sites in other parts of the cortex. But when you look at the actual CA fields, it doesn't look like this HM phenomenon at all. Instead, what it is something more like other parts of the limbic system, like hunger or thirst or sexual behavior or agonistic behavior, sort of, getting angry, getting afraid. It's just, you are here now. You're such and such afraid, you're such and such sexually attracted, you're such and such thirsty, and this is the position you're in at this moment right now, and this is the direction your head is pointed in. So that's what this circuitry looks like it's doing. Now, it's clearly got pretty close access, through the through the septum and the hypothalamus to all these appetitive signals. So it's not like it doesn't have access to it. But strangely, what it looks like is more of sort of like an internal, monitoring system. Now, obviously, if it's really helpful to have something that knows you're in this part of the T maze, because that's where the reward is. But it turns out it doesn't seem to be modulated much by whether or not the reward is there. So you can put the reward on the other side, and this place cell will still fire, even though the reward is not there. So the actual modulation by whether or not there's a reward there is pretty small compared to the modulation by what the place is. So that's the. So this did not answer your question, which is how does this work? Not only how does this work, how could you possibly construct a place cell? But there's another question: is like, how do the place cells get? What do they have to do with perirhinal cortex and storing long-term memories? There's various theories about this, but I would have to say the answer is it's not entirely clear. We know this place cell phenomenon is very strong and explains most of the variance in the firing of hippocampal cells here. We know that this retrograde memory problem, was actually not coming from the hippocampus. It was coming from the fact that they had also lesioned perirhinal and perihippocampal cortex. But, how does the whole picture fit together? There's several theories, but nothing that just blows you out of the water. Yeah.
Yeah, so I'm not gonna lie, still a little confused. Because based on like the rat in this scenario is in a brand new environment, correct? So how does it just like pick a place?
Well, that's a very good question. So when I talk next time, I'll talk about, once people saw this, they said, wow, this is totally cool. We can do thousands of experiments. Like, what if we put him in a square box? What if we take a round box and just make the round box bigger? What if we connect up two boxes with a door that he can't go through and we let him through the door? So they've done, or what if we slowly expand something while he's not looking? What if we move the cue card around when he's running around in the box? So they've done thousands of experiments to sort of see how, what the control of these place cell firings are. And it clearly involves some kind of memory, because if you take the rat out of the box for a week, and if you manage to record from the same cell, stick him back in, the place cell is still there. So it clearly has some kind of memory of, where the cue card is, or what the meaning of, where the place cell should be relative to the cue card. So it's not that there's no memory going on. There clearly is a form of memory going on, but the part that looks so different from HM is it's just like you're right here now. It's living in the present. There's no kind of access of sort of direct access of old memories, or thinking about old memories, or reinstating old memories. It just looks different. But yes, the question of how all of this works so well, it's not that well understood. But there's several theories about how you might get a place cell that I'll talk about next time. One involves an attractor network, like we were talking about way back in the Paleozoic at the beginning of the class. So, yeah, probably the biggest gap, we have a reasonable idea of how we could make a place cell. Probably the biggest gap, I would say, is how does this sort of place cell business somehow, relate to sort of long-term intermediate term memory, periurinal cortex? How does that whole thing work? And then how does the appetitive signals get in here? Because I didn't draw any of the connections, from the septum, and from the hypothalamus. They're coming in there. They're definitely getting into the hippocampus, no question. So hype. So hypothalamus, they're getting in there too. So how does all these appetitive signals interact with all these different modality signals, interact with this intermediate term memory, interact with the place cells? It's complicated. But we have some reasonably good localization, so that this kind of dumping the contents of current working memory seems to really involve this, perihippocampal, periurinal stuff. And then the CA fields have a form of a memory where, here's where you are in the particular environment, but they seem more like sort of online monitoring of where you are now. But we'll talk about a bunch of cool experiments they've done with place cells and head direction cells next time. Is there any other questions about HM? Or alas, HM finally died. So now they only take out ever one hippocampus. They never take out both. Okay, Stan, I think we're done there.